Hello friends, welcome back to my channel. This video is the second part of previous video where we had started the topic, that is microbial classification system. So in today's video we are going to discuss in details the nutritional classification of microorganisms and nutritional transport. So in the very first image which I have just put here for the display, you can see that I have just drawn the membrane, and I have just displayed the different channels or different transporters which are available, which are embedded in the membrane of the bacteria. And this here in this image, I have shown the active transport system where energy is required for the transport of nutrients across the membrane, and it can be symport, antiport, or uniport. We will discuss it in details. So if you have not seen my previous video, which was on the classification system of microorganisms, that is phenetic classification system, phylogenetic taxonomy, phylogenetic and phenetic system, and genotypic system. So you can see that I will give the link in at the end of the video. So let us start this video. So nutritional classification and transport of nutrients across the membrane of bacteria. So as we all know that every organism in this on this earth requires nutrients, but based on the different sources, they require carbon source, or they require the electron source, or they require the energy source, but not all, but not one at a time. Every organism require all these three: carbon source, electron source, as well as energy source. All the three are required for the growth of microorganisms as well as for us. So here, based on this, first, let me discuss the classification of based on the carbon source and electron source and energy source, and later on we will discuss all the three together, that how they are classified, and that is the important part which is asked in the question examinations of GSET or JAM or CSIF NET or other competitive examinations. Okay, so let us start this on the based on the carbon source. We have the organisms categorized into autotrophs, which requires the CO2 as the sole carbon source, or they can be heterotrophs, where they require organic molecules as the carbon source. Okay. Now, there are some organisms which have the potential or which have the capacity to utilize uh, both. Either they can use carbon alone, or they can use organic molecules depending upon the availability of the carbon source in their environment. Okay. So such are called the mixotrophs. Next, we have the electron source. So in the electron source, because it is also required for the growth of the bacteria for various energy generation processes and other things. So if the bacteria is capable to use inorganic molecules as electron source, they are called as the lithotrophs. Lithotrophs. Okay. Otherwise, they are called as the organotrophs, where they can use organic molecules as the electron source. And the third one is the energy source. So for this, from where they are getting the energy for the function. So if they are using the energy from, they're getting energy from light, obviously they are the phototrophs. Otherwise, some organisms which are unable to process light, or which are growing in the dark sea, or which are growing in the dark areas, dark regions, dark places, so they are the chemotrophs, where they oxidize the organic molecules or inorganic molecules, both, for the energy source. Okay. Now here you can see that now, as I had told, that organisms require all the three, not only carbon source, or not energy electron source, or not only electrons energy source or electron source. They require all the three for their growth, for their proper functioning. So now, when we club all these parameters, then we come up with the nutritional classification system, where we have the, you see here, let me just sorry, uh, yes. So see, if we have the as you see the name, photo litho autotop. So how it has come? Photo, that is the energy source is, if the energy source is light, it is photo. Inorganic molecule as the electron source, it is litho. And if they're using carbon source, CO2 as the carbon source, so photo litho autotrophs. Examples of this I will discuss more in the next table, but for now, purple and green sulfur bacteria and cyanobacteria are the examples. Examples are very important. You must know. I will give the names also in the next slide. Okay. Then second category is again energy source is light, but the electron source is not inorganic, it is the organic. So as the name organic, so organo, and the carbon source also is the organic, though hetero. So photo organo heterotrophs. Okay. What are these? Purple non-sulfur bacteria and green non-sulfur bacteria. Okay. Why non-sulfur? Because the electron source is different. Okay. Third category, we have the inorganic electrons energy source. So inorganic means chemo. Okay, so chemo electron source you see here, it is not organic, it is the inorganic. So chemo, litho, and carbon source is CO2. So auto, chemo, litho, autotrophs. You see here chemo, litho, autotrophs. Okay, and examples, there are so many examples: sulfur oxidizing bacteria, hydrogen oxidizing bacteria, methanogens, nitrifying bacteria, iron oxidizing bacteria. Okay. Next category, we have the chemolitho heterotrophs, and the examples are sulfur oxidizing bacteria. Sometimes they are also called as the mixotrophs, which I discussed in the previous slide. Okay, and the next one is chemo organo heterotroph. Okay, chemo organo heterotrophs. So in this, most of the non-photosynthetic bacteria, because it is the organic source are being used as carbon source. So non-photo, and the energy source is not light. So non-photosynthetic bacteria, including pathogens, fungi, many protista, and many archaea. So I hope this is clear. If not, again I have discussed it, but here I will emphasize on the example part, which is very much important. But because in many competitive exams, this used to be asked, like they may give the name of bacteria, and they may ask this bacteria belongs to which of the following nutritional system, or which nutritional classification system this microorganism falls. So you can see here, like cyanobacteria, nostoc, cannabina, or some algae, chlorella, spirogyra, they are in the photolitho autotrophs. Examples. Next is we have the rhodobacter, purple non-sulfur bacteria, rhodospirillum, green non-sulfur bacteria. We have chlorophaxis and chlorobium. These are all the photoorgano heterotrophs. Okay, then we have so many nitrifying bacteria there, like nitrosomonas, nitrobacter, thiobacillus, and bogatia, that is sulfur oxidizing bacteria. They are in the chemolithoautotroph. We have some sulfur oxidizing bacteria which are in the, you see, chemolithoheterotroph. Okay, chemolithoheterotroph. Okay, this one is chemolithoheterotroph. Then we have most of the pathogenic bacteria, that is E. coli, the lactobacillus, then yeast, mold, protozoa, amoeba, paramecium, they are the chemoorganoheterotrophs. Okay, so please remember the examples. These are very important, and in many competitive exams, this one is asked. Okay, now proceeding further. This was the nutrition classification of the microorganisms, where you must know the different classification system based on the carbon, energy, electrons, okay, and their characteristics, what they require, how they get, and examples. Now we move further for the transport of nutrients. Like, okay, the nutrients are available in the environment, from the surrounding environment. Bacteria or microorganism need to uptake the nutrients, take inside the nutrients. So how they do this? They have defined mechanism, okay, and they are not equipped with only one. They are equipped with multiple such mechanisms. Okay, so let us discuss on this. First of all, why transport is required? As I told that when the microorganisms are growing in the poor nutrition habitats, so they must be capable of uptaking the nutrients from the diluted solution into the cells against the concentration gradient, because it is not if they are not growing in a favorable condition, so they must uptake those nutrients for their growth and development. Okay, then these nutrient, as the bacteria is not a open system, it has a cell wall, it has a cell membrane, and the cell membrane is selectively permeable, semi permeable, so it will not allow anyone to come in or go out, though okay, so there must be some transport system across the membrane that will facilitate the movement of nutrients from outer environment to inside the cell, and At the same time, movement of waste of the metabolism generated by the bacteria from inside the cell to outside. Okay, so nutrient molecules must pass through the selectively permeable membrane, and this is done with the help of transporters. There are some important, some unique transporters which play a very important role here. And there are so many different transporters which we will discuss later on. Okay, now based on the energy requirement, the transport system can be classified into two. One is the active transport system and the other is passive transport system. So in the active transport system, ATP or energy is required, and it is the transport of solute molecules to the higher concentration gradient from the lower concentration gradient, and this is also called as the against the concentration gradient. Okay, and since it is against the concentration gradient, that's why energy is required in the active transport system. While in the passive transport system, since it is towards the concentration gradient, so no energy is required. It takes place easily. Okay, like passive diffusion and facilitative diffusion are the examples. Here you can see I have just summarized this in the table. Transport system, passive and active. Energy requirement in the passive, there is no energy requirement, while in the active transport, energy is required. Okay, then direction of molecules in the passive, where no energy is required, it is the towards the concentration gradient or down the concentration gradient. That is from higher concentration of solute to lower concentration of the solute. Okay, example, simple and facilitated diffusion. But in case of active, where energy is required, it is against the concentration gradient, that is from the lower concentration to higher concentration, because within the cell the components are required in the high concentration, and outside the inner cell where the bacteria is growing, there are chances, there mostly it happens that those nutrients are in very less concentration or a very diluted form. So from that diluted form, bacteria requires those nutrients to take up inside the cell. That's why it is requiring energy requiring process, and it is against the concentration gradient. Examples are ABC, that is ATP binding cassette transporters, and the group translocators. We will see in the image. Here is the image. I have taken this image from the Block Biology of Microorganisms 16th edition book, and which is published by the Pearson Press. You can also refer this book. This book is very good for the microbiologist. So you can read this content in Prescott as well as Block Biology of Microorganisms. Okay, so let us see the simple transport system where again all these represent, these are the active transport system. Okay, because everywhere energy is required. In the simple transport system, you see it is driven by the energy. Here you can see hydrogen from the higher concentration is coming to from outside the cell to the inside the cell, and sodium, it is going towards the from inside to outside. Okay, and the example of this simple transport is the lactose transport, where the lactose is transport from outside to inside with the help of enzyme lacpermase. Okay, and you can see here, you can see that lactose as well as the hydrogen both are coming together inside in one direction. Okay, lactose is also coming inside, hydrogen is also coming inside. So this is symport, two molecules coming together in a single direction, that is symport. And here this is the antipode, because two molecules passing through the channel but in the opposite direction. You can see here sodium is going outside and hydrogen coming inside. So this is okay. Let me just write here for your ears. Here two molecules are moving at the same time opposite. So this is the antipode. It is already written here. Okay, and this is antipode, and here it is symport. Okay, antipode and symport. In another active transport system, we have the ABC, that is the ATP binding cassette. Here you can see the molecule which has to be moved inside. First of all, it binds with a binding protein. This is the first component. Second component is transported substance, the substance which is to be transported. This is the second component. Third is the transporter itself. This is which transports. So it is the third component. And inside, when it comes, it is released by the ATP. The ATP is released, the ATP hydrolyzing enzyme. This is the fourth component. So four components are required in the ATP binding cassettes. You must require this. You must remember this, that how, and once it binds here, the entire channel flips and it helps in the transportation. Okay, and then we have the group translocation where more than one enzymes are required. Chemical modification of the transported substance is driven by the phosphoenol pyruvate for in this example, and there are many other examples. So you can see here enzyme 1, enzyme 2, and HPR. These are the components of the inside. Then this is membrane bound, this is transmembrane, enzyme 2C. Okay, and this is how the transportation takes place. We will discuss it in the later on. Okay, now I have just summarized this, the entire which we discussed just now in one table. I have tried to with examples. So you can see the transport mechanism simple. Okay, the simple transport mechanism where no energy is required. Again, the facility to transfer, again, where is no energy is required. And it is here down the concentration gradient. Okay, again, no energy required, down the concentration gradient. Examples: oxygen, transport of oxygen, carbon dioxide, sugars, amino acids, when it is in the very high concentration in the environment, so it can be taken up. Example: bacteria. Most bacteria uses a simple diffusion. E. coli for the lactose uses same facilitated diffusion. And characteristic: the molecule should be small, non-polar. It can, like here in case of facilitated, molecule can be large also, but it should be able to pass through the the channel. Okay, now these are the active transport system. In the active transport system, we have the it is required energy and it is against the concentration gradient. Okay, again group translocation, again it requires energy, phosphoenol pyruvate, and against the concentration gradient. And examples for which nutrients are transferred in this way: this is a sugar, amino acids, okay, vitamins. Example of the bacteria which are performing this, these are the, for example, in the Streptococcus pneumoniae, multiple transport takes place by the ABC system. In E. coli, phosphotransferase system is there, which transfers sugar by the group translocation system. And so this table is a summary. You can get everything here. You can just this will help you a lot in figuring out the answers of the questions which are asked in the competitive exams. So you can just take a note of this. Okay. Okay, don't be afraid. Here I have enriched the more information and with specific examples. And I have also here, you can see here in this, I have tried to give you more examples and a more elaborated way. Let me just, we have already discussed it, right? Simple diffusion and facilitative diffusion we just saw, where there is no energy is required. Most bacteria have this, and all. Example, which of the, like here you may find the question that which of the following nutrients are transferred by any of the method may be given, and you may be asked this question. Okay, so this table is going to help you a lot when you are going to face the examination. So please have go through this table very carefully. I will just discuss a few, like here in the active transport system we have the primary active transport and the secondary active transport. Okay, earlier I had not used this word. Now what is this? In the primary active transport, you will see it uses ATP directly from the for movement against the concentration gradient. Okay, but in the secondary, you see, uses energy from an electrochemical gradient. Okay, here ATP is directly used, but in the secondary, uses energy from an electrochemical gradient to move the molecule. Energy requirement you see here in this, in the primary ATP system, it is the ATP which is the energy is providing the energy for this. Where in the second case, the secondary, it is the ion gradient which is providing the energy. Okay. Okay, group translocation we have already discussed. Phosphoenol pyruvate system is there. Energy required in the form of PEP, alter and transports molecules simultaneously. Okay, we have another system, PTS, phosphotransferase system. Here again PEP is required, and you see here simultaneously transport and modify sugar using PEP. Okay, and where it is used in for the transport of glucose, mannose, or fructose by microorganisms like E. coli and E. coli and Streptomyces mutans. Okay, then ATP binding cassette system. Here, the active transport using ATP to move a wide range of substrate. Here, energy is ATP is used, and It includes various bacteria, various molecules can be transferred. Like for example, amino acids, peptides, and ions are transferred in this way. Then we have the siderophore mediated ion uptake. Siderophore mediated. Siderophores comes when iron comes into a seed. So you see, utilizes siderophores to scavenge and transport ions. Siderophores are what? These are the inclusion bodies which are present in certain bacteria. Okay, you may find this term inclusion bodies. So there you will find this siderophores. Okay, now here energy is not required because siderophores bind to the iron directly, and for iron uptake especially it is used. And for example, E. coli and Pseudomonas performs this. I hope this might be very much clear for you because this table is very informative, and I have tried to summarize the whole information in this single table. Okay, this is just a simple illustration of the symport, antiport, and uniport. You see, here one molecule is traveling in a single direction. So it is uniport, single molecule is transferred, no energy is required. And example of this uniport is GLUT1. Okay, you can have this example because it may be asked in the competitive exams. Second is the symport, same direction. Okay, you see this one, the same direction. Both molecules are transferring in one direction. No energy is required. And sodium glucose symporter 1. Okay, SGLT1. This is the example of this. And then we have the third example, that is antiport, where the two molecules move through the same transporter in opposite direction. Okay, it may or may not require the energy. An example is sodium calcium exchange, NCX, for this. So keep more get and find out more examples of such transporters so that you can get the answers in the questions. Okay, this is beyond these transporter systems. We also have secretion systems present in the gram-negative bacteria. Okay, this will be the part of another video that I will discuss later on. I have just shown it so that you can be sure that this is the earlier which we discussed, that is not the only transport system. There are the secretion systems also through which bacteria secretes the internal components, internal metabolites from inside to outside the bodies. And we will discuss later on in the other video where I will tell discuss that these uh, transport systems, secretion systems play a very important role in one of the phenomenon that you might be knowing, that is the anti-drug resistance, multiple drug resistance. So one of these are responsible for that. That's the part of another video that I will bring up some other time. So I hope you have enjoyed this video and you have learned a lot. So as I say always, if you got the information, if you liked this video, then please subscribe to my channel, like this, and share it within your friends and peers so that they can also get benefited from this video. So I hope you have enjoyed and you have learned the things. Give me a like and share it. So thank you so much for observing my video till last. Thank you so much. Thank you.